Inside every single living thing, there are these tiny microscopic machines working at just incredible speeds. They literally make life possible. They're called enzymes, and today we're going to decode how they work and, just as important, how fast they work. Okay, let's dive right in.
So let's start with something you've probably seen 100 times. You get a little scrape, you pour on some hydrogen peroxide, and boom, this crazy fizzing starts happening. Well, that's not magic, it's biochemistry happening right there on your skin. And it's the perfect way to kick off our story today.
So, who's the star of the show? The reason for all that fizz? It's this magnificent molecule right here, an enzyme called catalase. It's packed into our blood and pretty much every living thing, and its whole job is to protect us from a dangerous chemical that, believe it or not, we're making all the time.
So what is an enzyme, really? Well, think of it like the world's most efficient factory worker. It grabs the raw materials, helps them transform into a finished product at lightning speed, and then, and this is the key part, it's immediately ready to go again, totally unchanged.
Now, catalase has a very specific, very important job. See, our bodies naturally produce bits of toxic hydrogen peroxide. If that stuff were to build up, it would cause some serious damage. So catalase jumps in and just blitzes through it, breaking it down into harmless water and the oxygen gas you see as those fizzing bubbles.
Okay, so we know what catalase does, but how does it actually grab onto that hydrogen peroxide and snap it apart? Well, that brings us to the really elegant mechanics of how enzymes connect with their targets, which we call substrates.
For a long time, the thinking was what's called the lock and key model. Simple idea, right? The substrate fits perfectly into a rigid little slot on the enzyme. But, science moves on. We now know the induced fit model is way more accurate. The enzyme isn't rigid at all. It's flexible. It actually changes its shape to give the substrate a perfect snug hug. So it's really less like a key in a lock and a lot more like your hand sliding into a glove.
And this little diagram shows the whole dance in action. You've got the substrate, that's the S, and it finds its enzyme partner, E. They hook up for a split second, forming what's called an enzyme-substrate complex. That's where the magic happens. Then, bam, the enzyme releases the finished products and is immediately ready to find its next dance partner, totally ready to go again.
Knowing how it works is awesome, but scientists always want to know more. They want to know how fast. And that's the whole field of enzyme kinetics. It's all about putting a stopwatch on these reactions to figure out what controls their speed.
So how in the world do you clock an enzyme speed? Well, based on a classic MIT lab experiment, it's actually pretty straightforward. First, you mix the enzyme and its substrate. Then, you just measure how much product shows up over time. In this case, you can actually measure the pressure from the oxygen gas that's produced. And from that data, you can calculate the reaction's initial speed.
But here's the next question. How does that speed change when you have more or less of the ingredients? To figure that out, biochemists use this really foundational tool called the Michaelis-Menten model. It's a super elegant way to describe the relationship between how much substrate you have and how fast the enzyme can actually work.
And the model starts with a really intuitive idea. Let's say you're a baker with just a little flour. You can only make so many cakes, right? But if you get a big delivery of flour, or in this case, more substrate, the whole operation speeds up. More ingredients means more product. Makes sense.
But there's a limit. Think about our baker again. Imagine they're working as fast as humanly possible. If you keep dumping more and more flour on them, they can't bake any faster. The same thing happens with enzymes. At a certain point, every single enzyme molecule is busy. They are completely saturated with substrate, and they physically cannot work any faster.
And this absolute top speed, the fastest that reaction can possibly go, no matter how much substrate you throw at it, is a super important concept. It's called Vmax. It just stands for maximum velocity.
So let's lock that definition in. Vmax is the enzyme's top gear. It's the red line. It tells you the absolute maximum rate of the reaction when that enzyme is working at full 100% capacity.
But top speed isn't the whole story. There's another crucial piece to this puzzle, Km. If Vmax is about speed, Km is all about efficiency. It tells you how much substrate you need just to get the enzyme up to half of its top speed. Think of it as a measure of attraction or affinity. A really low Km means the enzyme is like a super strong magnet for its substrate. It gets to work really fast, even when there's not much substrate around.
Okay, so this is all fascinating theory, but why does it matter? Why should you care? Well, it matters because we've figured out how to harness the power of these incredible little machines in countless ways that really do impact our daily lives.
I mean, just look at this. The global market for industrial enzymes is gigantic, and the biggest user by a long shot is the detergent industry. Yep, those stain-lifting enzymes they advertise on your laundry soap, that is exactly what we were talking about. But you can see they're also used everywhere, making textiles, processing food, baking, even making animal feed better.
And honestly, the list just keeps on going. Enzymes make your fruit juice clearer, they improve the texture of your bread, they help bleach paper with fewer harsh chemicals, they're even absolutely essential tools in modern medicine and DNA tech.
So, where do we get all these enzymes for our laundry and food? Well, for the most part, we use biotechnology. We figured out how to harness tiny microorganisms like bacteria and grow them in these huge steel tanks called fermenters. It's a really complex process, as you can see, but it lets us mass-produce these molecular machines on an incredible scale.
You know, I love this quote. It says, enzymes can rightly be called the catalytic machinery of living systems. And that just hits the nail on the head. They are the machinery of life. Without them, all the chemistry our bodies need to survive would just be way too slow. Life as we know it just couldn't exist.
So from cleaning our clothes to literally powering ourselves, enzymes are the unsung heroes of the molecular world. And understanding their speed, their kinetics, lets us put them to work more and more effectively every single day, which really leaves just one question. What problem will these tiny biological machines solve for us next?